से वो ह्यूमन, एनिमल, प्लांट और उसको इफेक्ट करना शुरू किया। तो उस पर हम डॉक्टर सिंह और डॉक्टर काय, ये 3 लोग मिलके उसको बुक लिखने के लिए जा रहे हैं। प्रॉब्ली दोस नेक्स्ट 2 years में वी विल कंप्लीट दिस बुक। सो दैट विल, वी आर ट्राइंग विद दिस बुक टू डिफाइन द evolution in an with a very new perspective which never looked upon. So probably we will have a good concrete theoretical framework about the evolution after compiling this book. So this is the short story about this.
Very nice. Very nice. And then once Professor Kai shows up, our Hindi talk will stop, obviously. So this is a chance to to speak in Hindi, whatever we want to say. तो उसमें एक और बात यह है कि survival is one of, I don't know whether Shuowei what is going to do. So I say I'm not very sure. Because there are so broadly there are a lot of things to be said. But एक जो sociological या social ingredient एलिमेंट डाला जाता है, जो कि चार्ल्स डार्विन ने खुद नहीं कहा था, वो है सर्वाइवल ऑफ द फिटेस्ट। तो इसके 2 रूप हो जाते हैं, जो सर्वाइव किया वो फिट है, फिटेस्ट है वही, या कि जो फिटेस्ट है वही सर्वाइव किया। तो उसमें चीटिंग होती है एवोल्यूशन में भी चीटिंग होती है कि लोग, अब जैसे किसी ने, मतलब ऐसे शरीर ईश्वर ने बनाया, प्रकृति ने बनाया है, लेकिन आर्टिफिशियली लोग बंदूक भी बना लेते हैं। इफ समबडी हिट्स यू विद ए गन, दैट्स नॉट योर नेचुरल एवोल्यूशन, इट्स ए आर्टिफिशियल एवोल्यूशन। तो बिकॉज द पर्सन इन भारत की जो परंपरा है कि उसमें ऐसे कहते हैं ना कि राम ने रावण को कहा कि अस्त्र लेके आओ, तब अस्त्र शस्त्र लेके आओ तब लड़ेंगे। अगर कोई निहत्था होता तो उसके पे वार नहीं करते थे। तो वो दूसरे तरीका का एवोल्यूशन होगा, बजाय कि हम कहीं किसी बात रात में दिन में आग लगा दें, ये कर दें वो कर दें। तो आज का जो राजकुमार ये कह रहे हैं, द रीजन, पार्ट ऑफ द रीजन इज ऑफ कोर्स वी हैव दिस एक्सपीरियंस, बट द अदर पार्ट इज मेनी टाइम्स इन सोसाइटी ऐसा होता है कि जबकि आवश्यकता होती है, तो कहते हैं ना कि वो द नीड इज द मदर ऑफ इन्वेंशन।
आवश्यकता आविष्कार की जर्नी,
आवश्यकता ही आविष्कार की जर्नी है।
Necessity is the mother of invention.
क्या बोला आपने?
Necessity is the mother of invention.
Necessity is mother of the invention. तो
आविष्कार
तरह तरह से कहा गया है। उसमें भारतीय उसमें जो हमारे यहाँ के दर्शन है, उसमें ये कहा जाता है कि समय के साथ चीजें बदलती ही हैं, तो आपको आविष्कार तो करना ही होगा, क्योंकि नेसेसिटी आपकी बदलती जाएंगी, उस समय की चीज साथ चीजें बदल जाती। But so is here, and
yeah, I'm sorry I get late because I saw the 9:30 for some reason.
Yeah, that's what I thought. That's what I thought. But that's okay. You know, we said your name is Shuowei, which means he shows off. So we have to wait for him because he is a very popular and important and celebrity type of person.
Thank you. Yeah, I saw the poster say the 9:30, so probably because the time changes. I don't know.
Yeah. So they need to evolve to learn the time changes.
I respond to Dr. Kai your request of time. That
I say the 8:30, and when I saw the poster, it's 9:30 a.m. Eastern time. So
I think Rajkumar was thinking that you should know the mistake we can make. So
I'm sorry, I apologize, because when he called me, I joined me. So
that's what I also thought.
Yeah.
Okay. So
okay.
Am I supposed to introduce? Somebody is supposed to formally introduce him.
Do you have slides, Dr. Kai?
You
can.
I have the slide ready. So do you have the?
I'm on the line.
They will give you the permission to share.
Okay.
So we have in the audience, we have people who are in linguistics, philosophy, sociology. So it's a nice topic that you have. I hope you will understand this. Of course, there are scientists, there are molecular biology,
Okay.
there are and other people also.
So yeah, so maybe I using some lay language to explain things.
At least, somewhere you may have to translate it to others so that people can understand.
Okay. All right.
Okay, so he's ready now. Somebody is still introducing.
Share here and share this one, and I see if I can get to the. Everyone can see here.
Yeah.
Okay.
Okay.
Thank
hello.
you for.
I think they are still going to formally introduce you. I just told you your name is so. That means so off, but that's not what they're going to talk about.
Oh, but I'm show off. So sorry.
Okay.
Namaste everyone, and I, Dr. Alok Dwivedi, welcome you all in our November month KSAS talk, which we often organize every month and invite the scholars from different disciplines. Today we have here Dr. Shuowei Cai, a very eminent personality, and the faculty members who are associated with the INADS are familiar with him. Dr. Shuowei Cai is the professor of chemistry and biochemistry at the University of Massachusetts, Dartmouth. He received his BS degree in chemistry and MS degree in analytical chemistry from Nankai University, his MS degree in biochemistry from University of Massachusetts, Dartmouth, and his PhD degree in biological chemistry from the University of Massachusetts. Under the supervision, this is the more important thing and very valuable for us, because he completed his PhD under the supervision of Professor Balram Sinsar, the president INADS. He was the supervisor to Dr. Cai. Dr. Cai has been working on the botulinum neurotoxin for over 27 years and has published over 65 papers in peer-reviewed journals. His research interests are the biophysical, bioanalytical chemistry, protein chemistry, and pharmaceutical biotechnology. Today's topic on which he is going to speak us on evolution of microorganism and the lessons we learned. It's over to you, Dr. Cai. You have 35 to 40 minutes, and then there will be some questions and comments by the audience. Thank you.
Thank you. Thank you for the very nice introduction. And again, I apologize for being late here. Sorry about that. So now I can share my screen to go over again.
The screen is already on.
Oh, it's already on. I couldn't see.
So you just start maybe.
Just full screen.
Okay.
Yeah.
All right. So I can put my laser pen here. So today we are going to talk a very interesting topic here as the evolution of microorganism and the lessons we learned from that. So go to the next. So quick intro about what the microorganism there. I have a lot of slides, so maybe I will cut some because we have about 40 minutes here. And microorganisms are the lives that is too small to be clearly seen by the unaided eye. So many times we have to use a microscope to see the microorganism. That's why we call this is a micro, right? And the microorganisms, they can be single cell, or we call the unicellular, or the multicellular, or even the acellular, or liking of the cells. And the cellular, including the bacteria. Today we are focused more on the bacteria, and a particular bacteria is what we are working on as a Clostridium botulinum. And we also have some other things, including this cellular organism, and for the acellular organism, including the virus and prion as well. So prion is a protein which can also cause for the mad cow disease, right? And the virus, everyone know the virus is non-cellular, but they have to get into the cell to get into our host to multiply. So this is an organism and a biological entity here. We have cellular, we have acellular, and we are talking about bacteria today here. That is, they all in the very early, they come much earlier than our human beings. So the importance of these microorganisms, they are very diverse and they can found everywhere. They play a major role in our system there, ecosystem there, right? They role in the recycling things, and also they are the sources of nutrients, and they make our, they can produce our food and vitamins. That's all in the micro bacteria. They can do all these beneficial to our society. And they are very important to our human health. And as you can see the number here, so there are 39 trillion microbes in our human bodies, right? So that's more than our own cells. And they also have very diverse the genes. They are also, add them together is 500 times more than our own genes. So, and it provides tremendous benefit to our health. Also, they can impact our immune system and our behavior and much more, right? So you probably heard of those gut-brain axis, right? So that is, now they think that whatever our behavior may be controlled by all these microbials in our body. And majority, they are good for us. They can, in our guts there to produce the vitamins we needed, right? But some, they can cause disease. Only minority got disease, but majority, we can live peaceful with them, or they can live peaceful with us. And the oldest known bacteria fossil there, that's from the cyanobacteria, that's come from the about 3.5 billion years old, right? So that means they die in our Earth or in our planet for at least 1 billion years old, right? So, and all these plant animals, they evolved in the environment filled with billions of independent microorganisms. So that's why it's so important. And all our microorganisms, including us, have co-evolving with this community of the microorganisms living in and on our bodies. So that is the important thing it should know, and we should be very kind to those microorganisms. And I think they are kind to us as well. So the reason, the importance of the microbial communities in the health and the development of other organisms raises a rich set of questions for evolutionary biologists. I have to admit I'm not evolutionary biologist, but we are the protein chemists. We are working on the proteins which those bacteria produced. But that's also, I think Dr. Baron Singh also is very interesting in all these evolutions. So that's why we are studying all these things. And they put a selective pressure facing the plant and animals and their physical environment, diet, and interaction with other species, and those feedback to affect the evolution of our microbial partners. And they evolve very fast, and therefore they can make them the most diverse organism on the Earth. And the discoveries made in microbiology have proven to have universal biological significance, or maybe universal social significance as well, since we have all the, in the audience, we have the people with all different academic backgrounds, so that may also interest to them for this microbial evolution, because it's proven as a universal to us. So early organisms, they evolved from the RNA, so that's the original of the molecule, and that's mass fulfilled as a protein, and hereditary functions there, and we have the 16S ribosome. So ribosome is a cellular organism. They are synthesis the protein. So nowadays we are still using this 16S ribosome RNA to tracing our evolution trees. So that is very important here. So here is early microbial evolution. So as many people know, that the Earth, when they form early, we don't have oxygen, right? So that's why the early life based on the anaerobic and involving that. And you can see the Clostridium here at the base there, right? So it's very early, and that is a bacteria we are working on. So that's I will talking more about this Clostridium and how do we use the evolution tools to study this Clostridium here. But remember that's in the very early life base here, right? So, and they become the base there. And if you see, because all these, they will go to the different metabolism early on, our Clostridium is here because that is using the acetic acid metabolism, because they are eventually generate acetic and they don't have any oxygen get involved. And when they start to involve there, so bacteria can have the cyanobacteria, so they can using the photosynthesis, and they are produce the oxygen, and slowly we can get all this life get involved, which right now we have to rely on oxygen for ourselves, right? But originally all these ancient bacteria, they don't need oxygen and they just involved, and through the time they get into the photosynthesis, they produce oxygen, and eventually we get our mammalians, our animals, and our plants, and our human body as well. So evolution of cellular microbe is based on the mutation. So mutation become an engine for the evolution. So the new genes and the genotype, they get involved with all these mutations, right? So, and for the prokaryotic cells like bacteria, they don't have the nucleus. They're different from our human body, or the different from the eukaryotic cells. Eukaryotic cells, they have a nucleus, but prokaryotic cells, they don't have the nucleus. They directly have the cytoplasm. And this cytoplasm, the gene gets our chromosome or the gene distribution, they all happen into the cytoplasm. Therefore, it's easier for the microbial to get evolved, because they can take the gene from outside and integrate into their chromosome easily, because they are just into the cytoplasm. And that's not the case for the eukaryotic cell like our human cells, because we have nucleus protect our gene or protect our genetic information. But for the early life, the prokaryotic cells, they don't have that. They directly into the cytoplasm, and therefore they evolve faster than the eukaryotic cells. And you can have the site mutation here. We call that the point mutation, de novo point mutation, or you can have the gene copy or gene deletion, or we saw that the gene loss, right? So that's called the large insertion or deletion, or we can have the horizontal gene transfer. That makes things more efficient for the evolution, because they directly take the gene from outside their species and integrate it into their own genetic information there. And this mutation can occur neutrally or under the selection. And it is very rare that the mutations are totally unaffected by the selection. And the selection can detect those mutations. I want to emphasize here that selection didn't produce or make the mutation. The selection is detect the mutations happening. And from the selection, they can apply for the selection pressure and therefore they select the mutation which can adapt it to the environment. And the natural selection is thought to favor the lowering mutation rate. For example, you don't let your cell get mutate too much, right? So we know a lot of cancer caused by the mutation, and therefore the mutation is harmful for us. So that's natural selection is thought to favor the lowering mutation rate. But for the bacteria, they have the high mutation rate, and that's because the bacteria have the exposed to a strong pressure to have to adapt quickly to the environment. And also the bacteria are defective in their repair mechanism. For us, we have the gene repair mechanism, so which can help us to get a lower mutation rate. But for the bacteria, they don't, right? So that's why they constitutively mutate at a very high frequency. And they are all under the pressure, so that's why we say that bacteria allowed. So those are pressure or selection pressure, because they exposed to the certain stressful or growth limiting conditions in the environment, allow the bacteria to transiently increase mutagenesis, particularly when they are exposed to the pressure to adapt into the environment. For example, the bacteria you have very quickly, they can develop the antibiotic resistant strain, right? So that's because you put the pressure, you're using antibiotics, you try to kill them, you put the pressure on them, and therefore they can increase their mutagenesis rate and to adapt that hostile environment. And I already mentioned the selection not cause a mutation, but it detects mutations. During this mutation, they can use in the large insertion or deletion. They can insert large copy of the gene into their chromosome, or they can delete those. So that's we call that the indel, right? So the indel are ubiquitous for the bacterial mobile genetic elements. And the bacteria, they have the mobile genetic elements. Basically, they come from those indel, or they come from the horizontal transfer. And they could be deleterious, they could be neutral, or they can be beneficial. And sometimes those are large, or most of the time, the large insertion can cause a frame shift. What means frame shift? We know that's a 3 nucleic acid coded 1 amino acid, and that's every 3, right? So when you insert a large, they may cause this sequence frame shift, so they will cause their codon going to be changed. So we call this is a frame shift. It's not like the site mutagenesis, they only change 1 nucleic acid there, therefore they don't change that the 3 codon. But now if you insert large, if they are not the integral of the 3, they are going to change all these frame shift to change to the different amino acids. And the horizontal gene transfer is very important because microbial acquire the DNA from a variety of sources. They can from the other species rather than themselves, the distant species there. So they transfer in one of the that's because they become one of the major evolutionary forces and reshape the genomes throughout the evolution. And they can using the transformation. Basically, they take the gene into their body there. So those can be from the dead organism there. So, but they can also from the live organism, right? So they can get this to directly into from the medium get into the cytoplasms, and because again their genetic information stores directly into the cytoplasm. Therefore, once you have the free DNA, they can get into the directly integrate into their genetic code there. And you can get the co-conjugation there. So that means one-way transmission mechanism of DNA from one cell to another, while this sexual pellets by which DNA transported, and they can do the transduction, which means occur via the bacterial phage. So phage is a virus, right? So they can transfer this from the bacterial phage and into their cell there. One thing we think about how do they do that is nowadays we have those RNA vaccine or the DNA vaccine. They also need to transfer into our body. So that's what we learned from this, how they transfer DNAs, and that's applied to our daily life. Nowadays, we have those DNA, RNA vaccine. That's also we do the transformation, right? So we transfer those genes into our cell. We need to integrate that into our cell. And once inside the cytoplasm, they can be destroyed by the DNA degradation system because we think that's foreign things. We don't like them. And they also can persist as an autonomous replicant entities such as plasmid. And we are talking about plasmid later in our Clostridium botulism there. And also all or part of DNA can be integrated into the host chromosome. Both the plasmid and the chromosome, now they can express. So as long as they can escape this degradation process there, they will going to express their proteins into our cell, either through the plasmid or through integrate into the chromosome. And this horizontal gene transfer offers the possibility of quite drastic adaptation. They can have the intro or interspecific genetic exchange there. As I mentioned, because they can transfer gene from the, excuse me. They can transfer gene from the other species, right? So that's why they can have this intro or interspecific genetic exchange there. And we have all this gene duplication, gene loss, and also we have the horizontal transfer, as well as the satiation of the signal of the homolog. All these get into the evolution of our gene repertoire. So that's make our gene pool very diverse. And they play a very powerful evolution force. So they are a very powerful evolution force for the microbial evolution and also in general evolution. So those are plays a very powerful role there. And before we're talking about the Clostridium one, so we are going to talk a little bit on the co-evolution. So co-evolution based on their interaction, right? So we call co-evolution also the reciprocal evolution or interacting things. So that's basically not only for the microbial, maybe also for us, right? So basically when you try to deal with the different peoples, we have to interact them and we have to repeat reciprocal, and we have to co-evolve ourselves so we can adapt to the new environment there. And this co-evolution is one of the major forces shaping the diversity of life on Earth. So we have to co-evolve with all these microbials because they are all in our body, right? We just mentioned they have more. Number-wise, they are more than our own cells, so we have to adapt to them, and they have to adapt to us. So that's why this co-evolution is important. And how do we study this evolution of microorganisms? We mentioned the 16 as rRNA or ribosome RNA sequence there, and also the molecular biology play a very important role there. And with the new generation of the sequencing technology and also the bioinformatics. And in the next part, we'll talk about how do we using that in our group using this sequencing information and the bioinformatics to study those evolution, right? So that's provide very needed tools and have been widely used to study those microbial evolution. We just using the molecular biology and sequencing information and bioinformatics. Okay, So now we back to the Clostridium. So we just mentioned the Clostridium here. I think Dr. Baron Singh, I work in the Dr. Baron Singh's lab since 1995. And the Dr. Baron Singh is a leader in that, a world leader in that Clostridium botulism and botulinum neurotoxin. So that's why we get involved into the research on this bacteria. So Clostridium at the base of the evolution we mentioned earlier, very early life. They have a large, they are the obligate anaerobics belong to the Firmicutes phylum of the bacteria, and the most, they are the gram-positive, but they have some species is gram-negative, but most Clostridium species is gram-positive. They are very ancient, we mentioned. They are in our early life, and they have been in the planet for billions of years, right? So Clostridium and Clostridium tendinous belong to this group. So we started with the Clostridium botulinum and Clostridium tendinous. And now we like to give a quick intro about this Clostridium botulism. And Clostridium botulism produces deadly botulinum neurotoxin. So that is the most poison things in our planet. So that's very toxic. And Clostridium tendinous is a closer cousin of the Clostridium botulism, and they produce a tendinous neurotoxin. And there are 7 serotypes of the botulinum neurotoxin there, from A to G, and some they also produce as a hybrid. So for the A, B, E, they occasionally F, they are target our human, they leading the human botulism. And the type C targets most animals, including the avian. We have the avian botulism, fish, mink, cattle, and horses. And bound D often related to the cattle botulism. And ABC also can cause the equine or the horse botulism. And type E can also cause the fish botulism. And type G is interesting. We have not found any type G involved outbreak of the botulism. So basically, the A, B, C, D, E, they all can cause the botulism, either the human or the animal, but type G haven't caused any the outbreak. At least we have not found that yet. And through this evolution there, we know this is a very, the Clostridium itself is a very large family of the toxin there, right? So, and the botulism and the tetanus, they are only the part of that family. And the horizontal gene transfer play a very major role in this toxin acquisition. So originally, Clostridium, they don't have the, at least what the hypothesis is, or what we thought is, originally the Clostridium, they don't have this toxin production, so they don't, and they acquire this toxin through the horizontal gene transfer. At least we have the scientific basis to claim that. So the sequences comparison indicate that the Clostridium botulism strains, the bacteria, and the toxin producing gene or the toxin, they evolved independently of each other. So we have this bacteria and we have the toxin, right? And there are abundant genes for mobile elements surrounding the bound gene clusters. So that means all these may come from, not directly from the Clostridium themselves, they come from outside because they are the mobile elements, they can move faster, right? And this bound gene, so which coded the toxin, they are encoded as part of the conserved cluster, where they are found linked to gene encoding accessory or non-toxic complex toxins or complex proteins there. And in most sequences, the strain there, the bound gene cluster are located in specific position on the chromosome or plasmid. So they can either in the chromosome some strain in the chromosome and some strain in the plasmid. And we mentioned when gene gets horizontal gene get intake into the cytoplasm, their destination either be destroyed or they become the plasmid or they can integrate into the chromosome, right? So that's what we believe the toxin gene may come through the horizontal gene transfer, get into the genetic system there. And the multi-domain homologs of the bonds were also newly identified. So that means those are toxins not only in some Clostridium species, they also in some other separate classes of the Formicules family. So that's strongly support that the toxin gene may go through the horizontal gene transfer, get into the cell. And the bonds, the protein, the toxin is very conservative structure. Their sequence not less conservative, but their structure is very conservative. They have the light chain and heavy chain. So heavy chain responds to the binding to the cells. Here we have the binding domain there, and also the membrane transfer domain. So we call that the translocation domain here to help to process membrane into our cell. And once into the cell, the light chain, which is the zinc endopeptidase, they behave as an enzyme that cleave very specific. Remember, this very specific, that's why we call neurotoxin. They only target neuronal cells, and they only target the neuronal exocytosis system there. So that is a zinc endopeptidase. They only cleave the snare proteins involved, the neurotransmitter exocytosis. And you can see this zinc binding domain is conserved through the different serotypes, including the tetanus, and they belong to the glucine clan there. And this is a metabolic endopeptidase, or the metal-related proteases, and that's involve many other proteins, not just the protein here. But you can see this is very conservative. And the light chain of bond and the tetanus could be derived from some other family of the metalloproteases, just means the protein, metal-involved proteases, right? We call that the metalloproteases. They could derive from other metalloproteases. And this HEXXH, which is zinc binding domain, is conserved through in a lot of other monometallic metalloproteases. And their substrate, for most of the metalloproteases, their substrate is not that specific. They can cleave many other proteins. But for toxin, they are very specific. Why that happened? We think that's probably go through this co-evolution of the substrate and toxin. So basically, they have the co-evolution because this toxin like the neuronal cells, right? Because again, this the binding domain, they binding to the neuronal cells, they like the neuronal cells, and therefore, through this co-evolution, they start just to recognize their snare proteins, which in our neuronal cells and control the neurotransmitter exocytosis. And once this neurotransmitter get inhibited, they cannot release out. That's why we get paralyzed. And that's why we call this is a neurotoxin, right? Because this only specific target our snare system in our neuronal cells, and they inhibit neurotransmitter release, acetylcholine release. And therefore, we are get paralyzed. So basically, the neurotoxin just paralyze us, right? Once we get paralyzed, especially when our lung muscle gets paralyzed, we cannot breathe in, and therefore we are going to die. So the neurotoxin not killing us. The neurotoxin only paralyze us. And when they paralyze us, we cannot breathe in, therefore we are going to die. An interesting thing that the neurotoxin, they are not killing the cells. That's why with the time and with the supporting system like the ventilation machine there, we can get eventually recovered from the botulism. So that's a site topic about evolution, but we think that co-evolution play a very important role for the toxin to get specific to our snare proteins. And here is a cleavage site we mentioned very clear, right? So bound A targets SNAP25, and the bound C, SNAP25 and syntaxin, and bound E, SNAP25. Also, another serotype target is the VAMP, which is another snare proteins. But even they targeting the same substrate, you can see their cleavage site are very different, right? So that is the uniqueness of this toxin. And interest in the computer science or AI, right? So we're using all these bioinformatic tools, computer-aided tools. We can analyze all these bound sequences database, because from this new generation gene sequencing technology, all the
You're disconnected. He was criticizing AI too much, I guess. Yeah. Abhinandan, you have any comments on AI becoming more intelligent than we?
Sir,
It can figure out who is going to criticize AI, and then they just cut them off.
Right now, people are talking about new world order based on digital order.
Yeah.
This is digital disorder.
yeah, this is the right word, I think. Digital disorder.
Yeah, that's a good idea. Actually, that can
Yes,
be
sir.
measured by entropy.
Yes, Dr. Kai.
Raj,
Yeah.
if you, I think I could disconnect it from the Zoom.
Oh. So
you get me there.
yeah, you open the Zoom again and then.
Okay, yeah, I got it.
Okay.
sorry, so now you can hear me again. Okay, so
Yeah.
Sorry about that. I get disconnected here. So let me share. Okay. So before I get lost, I just mentioned this sequence there. So we're using this bioinformatics tools. We call that the BLAST, or basic local alignment search tool there, to analyze all our bound sequences there, because all the Clostridium botulinum genome gets sequenced thanks to all this new gene sequencing technology there, right? So we get all these strings there. So when we analyze them, we determine the so-called omega or value there, or also we call the DN over DS ratio. So that tells us the mutation ratio there. So that is evaluates the mutation rate. So DN is the number of the non-synonymous substitutions. So what?
Can you please do the slideshow?
Okay. All right.
Thank you.
Yeah, thank you. So what means using the lay language to say the non-synonymous substitute means you are going to change the sequence, the nucleic peptide amino acid sequence. And for the synonymous substitute, they don't change the peptide sequence or the amino acid sequence. They only change the nucleotide sequence, but remember we have the 3 nucleotide coded 1 protein, and they have the different codon can code the same amino acid, right? So for the synomics, they don't touch our protein sequence. For the non-synomics, they're going to change our protein sequences. So therefore, we are using this number of the non-synomics substitution versus the synomics substitution to determine this omega value, and omega value can evaluate the nucleotide substitution events there. And when this ratio here, omega value dn over ds, or also we can call Ka over Ks, right? So the Ka is the total number of the non-synomics mutations. So that means total number, the mutation event that change the amino acid sequences. And here Ks is synomics events. That means the total events, even though they change the nucleotide sequence, but they don't change the amino acid sequences. So you keep the same protein, even though their sequence are different, right? Even though their DNA sequence different, but you keep the same protein there. So this ratio greater than 1 means your protein sequence get changed. And if 0 means they don't change the protein sequence. So that's why when they're greater than 1 means they have the positive selection existed. You have the positive selection pressure there, like our antibody, right? So when you see the foreign things, our antibody change the sequence of which they can bind to create the antibody which can bind to the harmful things, so they can help us, right? So that's for the antibody, they have very large positive pressure, therefore their omega value greater than 1. When they are equal to 1, that's equal, so there's no selection, then neutral, right? And then when they're less than 1, that means they will keep the protein all the same, even though they could change the DNA sequence, but they keep all the protein the same. So we call this is a purifying selection or the negative selection. If you're looking for all these sequences there, just using the diagonal here, right? So they are at A here, omega close to the 0. So that means very stable sequence. Amino acid, almost no change there, right? So the 4 strings available, they're amino acid almost identical. So omega close to the 0. And for the toxins themselves, you can see within their species, of course, across the species, they are changed, right? But within their species, for example, type B there is 0.4, type C 0.8, they all less than 1. So it means they are all pretty much stable or conservative. But when we're looking for the different subunit there, we mentioned that the light chain, heavy chain, right? So light chain, if you're looking for the light chain, type E have very large positive selection pressure. So that means the type E, their light chain sequences, amino acid sequences is changing, right? So that's a type E here. So that's for the light chain omega value. And for the heavy chain respond to the receptor in the transformation, the translocation, right? So for the heavy chain there, interestingly, is a tendinous neurotoxin. Tendinous neurotoxin, they have very large omega value. So that means they enter the positive selection pressure there. But in either way, the omega value for A is very stable. So that's very interesting. So therefore, there's no speed up evolution pressure there in either among the different serotypes or the same serotypes. If you remember, we're looking for the food chain here, right? They're all less than 1 here. They're all less than 1 across the species. And within species, they also less than 1. And of those, the type A is more conservative because they are almost close to the 0, right? And when we see the different subunit there, they are quite different scenario. That's type E light chain and the tendinous heavy chain. They have the faster evolution rate than other sequences region. And bound A, again, we mentioned that seems to be the most stable neurotoxin during the molecular evolution. And we are using also the sequencing tools to establish this phylogenetic tree. What means phylogenetic tree? Phylogenetic tree is a evolution tree there. And you can see here the phylogenetic tree there. Type A is ancient, right? So they start from C and the D, and A is older than the E. E is the younger one, right? So E is the younger one, and therefore what we see there potentially correlated to the type E here. They have the light chain there. They have the large positive selection pressure. So that means they still keep evolving, right? So type E still keep evolving here. So type E light chain has faster evolution rate than other sequence region of bond, and therefore from the phylogenetic tree there, they also the youngest one. That means they are the latest based on this phylogenetic tree there. And the entire sequence region of bond A has been experienced very strong purifying selection pressure because their omega almost zero. And that's also telling us something, right? So we mentioned this toxin gene may be integrated into the Clostridium strain later, right? So separately, I should say, not later, but separately. So bond A is more critical to the surviving host, the survival host of the bacteria. That's C. Clostridium botulinum. So think this way. So if you change some sequence there, so they may toxic or harmful to the bacteria, and bacteria cannot survive. Right? So that's why we say this Clostridium toxin A here, they experiencing a strong purifying selection pressure there, because if they change, they will affect their life cycle of the bacteria, right? So that's why we say the bond A toxin may play a more functional role in the life cycle of the bacteria than other cell type toxins from this evolution study here. And I want to spend next 5 minutes quick because I think I ran out of time here. So to see the correlation between this botulinum neurotoxin and their neuronal substrate, right? So neuronal substrate, we say the type A is SNAP25, and type E, type C, they all SNAP25, and C-taxin is type C there. And we are using the multiple sequence alignment to analyze all the record of the substrate, and we calculate this conservation index. Conservation index tell how fast this SNAP25 or the substrate change in the cleavage site. So that's a cleavage site, P1 and P1 prime. So conservation index telling how fast these cleavage sites get evolved or they change, right? So without going these technical details there, so we're just looking for the number. The larger conservation number means the more conserved of their cleavage site of the substrate. And opposite to the toxin themselves, you can see the substrate for type E is more conserved than the type A or type C, right? So because type A and type C, they are ancient, so they are more stable. That's what the hypothesis here, or that's at least what we try to explain this phenomenon is. Type E slowly show by far the largest conservation index, and potentially because type E itself is very stable, is keep evolving because the toxin, if you remember the light chain, they are evolve faster, right? So therefore, their substrate gets conserved. And also type A here, they are the slowest evolution, right? Because they are more conserved. Therefore, their substrate going to adapt to them. Therefore, type A type A substrate cleavage site, they mutate faster than the type E, right? So that's where we get here. So that means the bond C also have the different things, but bond C just between the type A, because bond C, remember, they have two substrates, syntaxin and the SNAP25, right? So therefore, there's lower than the, faster than the type A because they have the two substrate there. So from here, what our conclusion is, the order of the conservation index reversely match their phylogenetic trees there, right? So that means the type E there, their substrate is more conserved. And type A sequence is conserved, their substrate is not conserved. So their correlation there telling us they may have some co-evolution relationship. And the type E, because they conserve, they evolve fast, therefore their substrate is conserved. And type A, they couldn't involve themselves, therefore their substrate is involved faster than the type E substrate. So what we learned? We learned basically all the synthetic biology learned from the evolutions. So synthetic biology is a field that leverages engineering principles to design and build a new biological system faster. They don't go through the nature. That's why we call it synthetic, right? And basically, they learned from the evolution and they mimic evolution and they apply the pressures during this evolution there, and therefore we accelerate this evolution and produce this evolution in our lab setting and create a new biological system there. So that's what we learned. So we call this is a systematic evolution ligand through exponential enrichment. We call SELECT. In this case, we put the random, instead of mutation, we put a random sequence there and we select through the target. And from there, we do the PCR and amplify and again through the different cycle there. And what we did here, we select the aptamers, which is a nucleotide. So that's a nucleotide library, right? So we select this nucleotide antidote against botulism. And we found several things, our labs found several aptamers which can detect the neurotoxin and also can inhibit neurotoxin enzymatic activities there. So that's what we published through this SELECT process we learned from the evolution, right? And another things in our lab we do is the phage display. Phage is a virus, right? So we put a random library into the phage there. Again, through not mutation, but through the randomized sequence there, right? We put a randomized sequence there. Then when we do the select, so we binding to our target. And again, the binding not cause the mutation or not cause this randomized sequence, but rather than detect, right? When we detect them, we select them and we keep using stringent selection pressure there. Eventually, we get this peptide sequence from the phage display. We call that phage display to strong inhibiting our light chain or bond A there. And some other things, now the more developed method is using the phage assistant continuous evolution. Again, that's evolution. We put the phage back to the bacteria and we're using the aeropro PCR. That means aeropro PCR means our polymerase means this polymerase can generate a lot of error. And during their PCR process, all this error we consider as mutation. And when we training them, you can see from this paper published a couple years ago in Science magazine there, they can reprogram the specificity of the neurotoxin just by the co-evolution, right? Co-evolution are the artificial system through the substrate. So I conclude my talk here as what we learned. So nature is our best teacher, and we have learned so much from the microbial evolution, from the fundamental evolution to apply to the synthetic biology. And also this is a fundamental principle of nature, and therefore the evolution adaptation have the implication beyond the biology. For those people working in the AI learned from the evolution and adaptation there, and they're using the AI, using this principle for the AI algorithm to help to involve all these programs. So that's basically is a universal, right? So evolution principles here is a universal, including the nature and also social science as well. For the human being, we need to involve ourselves and also to adapt to our environment, including the human-human interactions, right? So that's called the human skill there, right? So that's, I think that I conclude my presentation here, and I'd like to give thanks to Dr. Balram Singh, which is I'm working with him. It's my pleasure to working with him as his student first and as a colleague later for almost 30 years. And our graduate student, Alex Chang, working on the evolution of the Clostridium botulinum, as well as the silax and the perpetual Genadarin, working on the silax, and Winston Cho working on the phage display, and Dr. Raj Kumar also working on the evolution part, and Dr. Shalim Mello helped us to working on the phage display. So thank you very much. And any questions?
Great job. So I have to go to another meeting, but I really enjoyed talking. One question, very quick question. Can we, and I asked this to Raj Kumar, can we tell how far in number of years between type A and type E?
I think that's from the phylogenetic trend. You can approximately count it that.
Yeah,
Not
I mean, that would be a good question to know.
Yeah, because there's nothing exactly for the evolution here, right? So you cannot exactly answer how many billion years, but maybe we can, through the distance here, try to figure it out, approximation.
Okay. All right. You guys can continue. I got to go to another meeting. Thank you.
Thank you, Darcy. Again, I apologize for the come too late.
That's no problem.
Yes, Raj. You mute, Raj.
You are muted. So please stop sharing then.
Oh, stop.
Add spotlight to both.
All right. Thank you.
Sorry for the confusion about the timing, Dr. Kai.
No, it's my fault. I should double confirm with you because I earlier mentioned 8:30 and now I see 9:30. That's all my fault. No worry. I apologize again.
Okay. So my question is related to, because I want to use this opportunity to ask you a question about, you talk about the horizontal gene transfer. Horizontal gene transfer is very, very, very rare between bacteria and human or eukaryotes.
Yeah.
So normally horizontal gene transfer happens when you have a virus, and the virus has the integrase, because human system does not have a active integrase in their gene.
Correct.
The 1.4% gene which is active, they don't have integrase there, but they have an integrase type of sequence in the dark gene. But we don't know, still don't know when they are getting active, that thing. So normally when you have a virus, for example, and those viruses are also very small amount of, not all viruses have integrase. So like for example, HIV virus has an integrase that can integrate their DNA to our genome. But when you talk about the bacterial gene, normally even if they enter the eukaryotic cells, they cannot integrate into your genome. So how they will, in the absence of virus, they cannot integrate their gene into the eukaryote cells. So how they will contribute towards evolution, bacterial genes?
I think bacteria is different. So bacteria, because they don't have the nucleus there, so that's easier to get the horizontal transfer. They can even take whatever DNAs or the nucleic acid into their cytoplasma, and they start to do the horizontal transfer. For the eukaryotic cells,
bacteria to bacteria is okay, but bacteria to human, I am talking about the bacteria to eukaryotes.
Oh, from the bacteria, the eukaryotic world, I think that's, you make a very good point, because eukaryotic, if you think about the eukaryotic cells, they are evolved later. Now they have much better protection system. That's why we have the nuclear there, and it's very difficult to get the integration into the horizontal transfer, as what you mentioned. And that's why the virus play a very important role. And the virus maybe, I think, from the evolution point of view, virus is later than the bacteria, and the virus sometimes considered just as some degraded bacteria things there, right? So that's why the virus play a intermediate role there to get integrated into the eukaryotic cells. And now, with once we learn all these things, and the more we learn, now we have artificial things rather than virus can help. But naturally, I think the virus play a very important role. A lot of our foreign genes come from the ancient virus. A lot of virus gene already integrated into our chromosomes. And if you remember, that's the controversial things about the COVID RNA vaccine, the people afraid that the COVID gene gets integrated into our cells because that's directly delivered into our cells. Once the genetic system gets into our cells, eukaryotic cells have much better protection system there. And many times all these things maybe become the not get expressed into our system, even though they integrate into our system, become the silent gene rather than the expression genes.
Okay, and in addition to this question, I was thinking that normally the bacterial system affects the eukaryotic system. So our gut bacteria affects us a lot in our physiology. So I was thinking probably the genetic variations is not that important, or for the evolutionary perspective, but the protein they secrete, that they can change the physiology of eukaryotic cells. For example, that this Clostridium botulinum, that not only cleave the SNAP25 or the snare protein, it changed physiology also of the neuronal cells. So that probably maybe some kind of epigenetic kind of things which initiated by bacteria, and that changed the eukaryotic gene expression of some of the genes which are not known. So can you please on that?
Yeah, I think your question is maybe not the bacteria themselves, but they change our behavior, and they are. Can you repeat your question?
So I was thinking, Miss, normally we always go by the genetic mutation and genetic changes and other things, which is trigger normally the evolutionary changes in any kind of organism or a species. But for example, I was giving example about botulinum toxin, which not only cleaves the snare protein, but it's changed the physiology of the neuronal cells. And that change, after some times, it triggers some epigenetic change in the genome of our system. When we have a data about microarray, the microarray data suggests that those genes are also expressed, which are not generally expressed in normal neuronal cells. So that trigger that questions that probably not only the genetic change are important for the evolution, there are some trigger from the protein which bacteria normally secretes to the eukaryotic cells, they can also instigate some changes in the genome.
Yes, I think definitely. And also I think that if you think about that toxin there, right? So just take toxin as example there, right? So they are not only change, cleave or inhibit the neurotransmitter release, but as us, we also have to be adapt that, right? So adapt that toxin be cleaved, we get paralyzed, and we also generate a lot of signal there. So that's basically is kind of the co-evolution there. And also those things will trigger maybe not in our genome system per se, or any eukaryotic cells genome system per se, but it's an gene expression level, the epigenetics, right? So they are going to change our expression pattern there. That's why say you are the Raj Kumar or you are the same Raj Kumar, but now because some this beneficial bacteria in your gut better than more than before, now they may trigger your epigenetics and your expression pattern will be different from the old time you're Raj Kumar, right? So now you become behavior or you get much better there. So, or you change your behavior there. So that's why this behavior change and epigenetic also can carry it on. And I think that's why we have this beneficial microbial basically is communicated to us or communicate to any host, I should say that. So that's, we are get a co-reciprocal or the co-adapted to each other and to good or to bad, for good or for bad, right? So for the bad bacteria, pathogenic one, that's probably for bad and maybe eventually kill us. And for good bacteria, they may be good. We have the peaceful and also they have the much beneficial impact on us, right? So, and we just change us become better and healthier. So that is my take from here. I don't know if that's answer your question or not.
No, thank you, Dr. Clay.
Yes.
Thank you for such a nice presentation. Actually, I have one question. If you go to the evolution, the bacteria evolve very, very early than the host cells, host organism like mammals and human and all those things. If you go to the Nike bacteria, this is Clostridium found in the soil, and the host basically, have the different Nike. If compare the Nike. So what selection pressure or evolution work, why this bacteria developed this special mechanism to kill a different host from the different Nike?
So I'm sorry. So I think your voice get cut off. So can you just repeat your last question there? So what's the? I'm sorry.
Yes. My question is that there is, if you go to the evolution, the bacteria came very earlier than the host. The bacterial host is, the mammal like monkey, animals, birds.
Right.
In evolution, it come very, 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 very late in comparison to the bacteria in evolution. So why? My question is that why this means if you compare the Nike, bacteria have the different Nike than the host. So, but why would soil bacterium, this bacteria evolve such a powerful neurotoxin to kill the specific host in evolution?
Oh, why they killed? Actually, I think most times they probably just want to be peaceful, right? So they don't want to kill, because if you kill your host, now you don't have place to live anymore, right? So that's why, but majority is not pathogenic bacteria. They are helpful for their host. That's for the majority. And when you're talking about those bad guys, they kill the bacteria, that's maybe under, well, that's only what I think. That's maybe under the pressure that's the they have to survive themselves. So in order to kill people there, right? So for example, they want to get the food, or that's why we have the war today, right? So major thing, we should be peaceful. We don't need the war, right? So, but now the same thing for the bacteria. If they have some other, those other bacteria, they may take more resources from them. Now they probably want to take more resources and eventually harmful to their host. When they harm for their host, they may also get eventually killed, right? So that's why if you think about this, the COVID give a lot of the information there. And you remember initially this COVID virus is very deadly. They kill the people and maybe not very good to them. And now they start to adapt it. When they adapt it, now they just make you a little bit sick, but we can peacefully, at least they can survive also from you, as long as you are not be killed, right? So that's why now become less and less lethal. So I think that's the bacteria or the microorganism in general, they also involve, try to avoid these suicidal things, right? Because when they kill the host, eventually they are also commit as a suicidal event there. They be killed as well, right? So that's why this evolution also proved that, especially for the COVID study there, we see that now they become less and less lethal there. So I hope that's answered your question.
Yeah. And second question is that because generally nowadays the environment changing very fast. If you compare with the bacteria, the human develop very sophisticated things like the sterilization process, food processing, which sterilize the food from the bacteria and develop the drugs. So what do you think this Clostridium develop new type of toxin in future?
I think we can learn a lot of things from this botulism, right? So first of all, as you mentioned, they are evolve very fast, and that's why we learn that from the synthetic biology there. So we can make artificial things there, just mimic them to accelerate this evolution and select the drug targets. And in our lab, we select the drug targets either from the aptamer or peptide phage peptide there to inhibit this toxicity of the toxin and try to find a cure for the botulism, right? So that's here we learned that from the evolution, and we press the artificial pressure there to accelerate this selection process. Because if by nature you need probably billion and billion years to get a strain get selected, but here we can do much faster in lab. So that is one thing we learned. And another thing we learned is you said that the environmental change so faster, right? Even for our human being here, right? So maybe, but our human being, remember, we also evolve and adapt. We try to adapt this change. And remember, at the very beginning, the planet is very hot, and the very early life is come from the thermophilic bacteria, right? So that they can tolerate high temperature. Right now, we are very comfortable. People talking about the global warming or climate change become warmer and warmer. Actually, that's right now is much cooler than the beginning of the planet or beginning of the life, right? So I think once the climate change, we as a human being also continually try to adapt the environment and try to evolve. So that's what another thing we learned from the microbial evolution.
Yeah. Maybe in future we see new type of toxin.
Thank you.
Yeah.
Dr. Cai, I have this little question that, in the COVID times, I have read this small article that was stating that using sanitizer like alcohol every now and then, it is killing our skin bacteria, and that is directly affecting our gut health, because obviously what the bacteria that are going on our skin when we are eating using our hand. They are going inside. Biotic, I guess. Right. So if we like, how can we in scientifically, like from microbiologists, is there a correlation found that eating from your hand can increase your immunity or improve your gut microbiome?
That's interesting question. Thank you. Yeah. So I think, first of all, I want to mention that majority of the microbial is beneficial, not pathogenic. Only the pathogenic there. So that's why the people try to clean their hands and using the alcohol to clean their hands and try to kill the bacteria there. Many times it's try to kill the pathogenic bacteria, but in the meantime, you also kill the beneficial bacteria. So that's why now we have the probiotics. You just eat the bacteria, right? So that's good for your guts there. And your question is using the hands to eat the food there. And our hands also actually have many good bacteria and occasionally bad bacteria there, right? So that's why when you're using the hands to bare hand to eat food, you just want to keep those good bacteria and avoid the bad bacteria. And it's difficult to see which good and which bad on our hands. So that's why the most health professional recommend you wash your hands before you eat. Even you wash your hands, and then you can use the naked hand to eat food, right? But also, I think we also have this immune system there. So if you become too clean or almost get the OCD there, operation there, now you probably get a lot of allergy there, right? So what they mentioned here in the US, there more people get asthma or the allergy than the other countries, or especially other the third world countries. And they think that's potentially because these countries, they become too clean. And now you not get exposed to those bacteria. Even sometimes they are bad, you can have the immunity and eventually it's become not harmful. So that is a co-evolution. We try to co-adapt to those bad bacteria. We have our immune system, we try to kill them, and eventually we don't care of them anymore, right? So now, because when you become too clean, so that I'm not advocate that you don't wash your hands before you are eating. I'm just saying you cannot go to too extreme. And that's why they mentioned that after the COVID in this country, we have the RSV outbreak, we have all the different diseases, and that's because the people is too clean. They always wear their mask, they clean their hands for 20 seconds before you eat, and all these things make our immune system not exposed to those microbial anymore, and therefore very easy to get sick. You lose your immunity to those bacteria. When you see first time, you get sick again, right? So that's why I think you don't go to extreme. You still have the good practice as what your health professional telling you, to wash your hands before eating and keep the good hygiene, but also you don't become OCD to become, otherwise you will also not be healthy, except you are the immune compromised person. For example, we have the person have the chemo, their immune system is low, they cannot respond. So that's why they have to keep the clean all the time. They have to stay in the clean room, right? But others, we have our own system to deal with those things.
Okay, thank you. I'm a cell culturist, so I have to rinse my hand now and then. That's why I ask you. Okay, so any suggestions to improve your gut microbiome, vegetables, or food stuff?
Well, I think the best thing is you have the better diet. So because you can eat those probiotics, you can have those probiotic supplements there, but they can only survive certain times. And compared to our guts, we have the 39 trillion there, right? So you only eat some billion there, so that's really not as helpful or not very helpful. It may have some help. But if you have keep the healthy diet, therefore you can generate all your, those are good bacteria will like your guts and they will stay there, right? So they mentioned that for people, for the vegetarian people compared to vegetarian people and the meat eater people, and they analyze their gut microbial and they find completely different. And most of the vegetarian people, they have much more beneficial microbiomes in their guts than the people eat a lot of red meat, right? So that's why the healthy diet is more important than you just taking the probiotics. So that's, I think, just keep your healthy diet will help you have the good microbiome in your guts.
Okay, thank you. Thank you, sir.
You're welcome.
If there is another question, so we can take. I think no.
Okay,
So
thank you, everyone.
You presented very well, Dr. Cai, and you were not alone here because Dr. Singh and Dr. Rajkumar was here in your team, and you all are working on the same projects, and you mentioned the whole journey on which you are working here, and you are very familiar to us, and you are very close to us, INADS and the whole team. And again, we will invite you to start the further developments on this issue, because you three all are working on it, and our INADS KSAS team is also working on it. So thank you very much for accepting our invitation and for beautiful talk. And thank you all for joining us. It was a little bit misconfusion due to changing the time zone in India and US. So we started early, then Dr. Cai came later, and we
I'm
mixed
sorry.
it.
I
And
apologize.
there's some of our audience are now joining because we pointed the wrong detail in announcements. Further, we will take it all the things in our mind, because few days ago the time zone had changed, and this was that happens. So thank you all for joining us in next segment of KSAS talk. Probably this month again we will join with another scholar for another topic. We have some suggestions also. We are working on that. That we can do some panel discussion over the topics which we have got some suggestions. We will definitely work on the suggestions, and your suggestions are very beneficial to us to improve our journey KSAS talk. Thank you very much. Thank you all. Namaste. Pranam.
Thank you, everyone, and I apologize again.